Hello everyone, welcome back. Today I am here with one presentation on cell membrane. In this presentation I will discuss about the basic introduction about cell membrane and some basic structures of cell membrane. In next slide I have one animation that will make you clear about the basic idea of cell membrane.
The plasma membrane is made up of several organic molecules. I will concentrate on two of them in this presentation. The first molecule is a lipid. It is called a phospholipid because instead of the usual 3 fatty acid chains, there are only 2. The third bonding position is with a phosphate group. The phospholipid molecule is divided into a head region and a tail region. The head region is the glycerol and phosphate group. The tail region is the fatty acid chains. The head is hydrophilic, attracted to water. The tail is hydrophobic, repelled by water. It is this property which allows for the unique molecular alignment that makes the plasma membrane. If a droplet of phospholipid was put into water, a mycele would form. A mycele is a sphere with the hydrophilic heads on the surface, the tails inside, away from the water. Our bodies are made up of approximately 60% water and trillions of cells. The water can be found inside the cells, outside the cells, and in the bloodstream. The location of water and the phospholipid, hydrophilic, and hydrophobic ends enable the membrane to form a bilayer of lipids. The currently accepted model of the cell membrane was developed by Singer and Nicholson in 1972 at the University of California, San Diego. Pictured here, the plasma membrane looks like a sea of lipid with icebergs floating in. The membrane is very flexible and has a fluid motion. This design of the plasma membrane is called the fluid Mosaic model. Quite often, these proteins have sugar molecules attached to them, forming glycoproteins. These molecules extend from the membrane surface and act as an identification marker. The proteins are used by the immune system to identify a cell as being self, or foreign called non-self. Each individual has their own biochemical makeup. In other words, each human being has their own set of cellular proteins. But if a cell comes into the body that has a different set of proteins, whether it is a bacteria or an organ transplant, the immune system may identify it as self or as non-self and attack. In order for the cell to survive, many different materials must be able to cross the plasma membrane. When water passes through a cell's membrane, the type of diffusion is called osmosis. When solute particles pass through a cell's membrane, the type of diffusion is called dialysis. Before looking at this movement through the membrane, it is important to understand the movement of molecules in general. Atoms and molecules are in constant motion. This random motion will disrupt the cluster rather than maintain it. This motion is referred to as Brownian movement. This motion is always present in some degree, in gases, in liquids, and even in solids. For instance, if a bottle of perfume is left open in a room, eventually, no matter where you are in that room, you are able to smell the perfume. The molecules of air hit the liquid, causing the liquid molecules to move into the air. At the time the bottle is first opened, there is a high concentration of perfume molecules in the bottle, a very low concentration of perfume in the air. As the perfume molecules are bombarded by the air, the concentration of perfume is dispersed into the air. In other words, the perfume is moving down a concentration gradient, high to low. This movement is called diffusion. It uses Brownian movement. Now let's take a look at another example of diffusion. In this aquarium of water, we drop some blue dye. As the water molecules move, they bump into the dye molecules. The dye molecules, at first in a high concentration, start moving and bumping into each other and the water molecules, moving into the area of lower concentration. As this movement continues, the dye spreads out and equilibrium is attained. Equilibrium is equal concentrations of the dye and water in every area of the aquarium. Remember, Brownian movement continues even after equilibrium is reached. Although extremely thin, the plasma membrane separates the contents of the cell from the environment. The membrane has several functions. This video concentrated on the movement of materials through the plasma membrane. Remember that this membrane is selectively permeable and can prevent materials from passing through or keeping materials in, and it will permit, even facilitate, the entrance and exit of materials. Materials may move through membranes passively or actively. In this video segment, passive transport, which uses Brownian movement, was introduced. Passive transport includes diffusion, defined as the movement of materials from a region of high concentration to low concentration. Two specific forms of diffusion are osmosis and dialysis. These processes are essential to the survival of the cell. Nutrients must enter, wastes must exit. The structure of the membrane allows for the processes to take place, which enables the cell to live.
Firstly, a plasma membrane is a membrane which forms a boundary between the cell and its exterior environment or external environment. और ये काम करती है एक बाउंड्री की तरह जिसमें जो कुछ एक स्पेसिफिक चीज़ें हैं वो एंटर करती है विद इन सेल और एग्जिट द सेल, and that's why all the biological membranes are selectively permeable. In addition to this, in case of eukaryotes, we have some membrane bonded cell organelles which controls the flow of material within the cells. Basically, plasma membrane is a protective covering which is made up of lipid bilayer and proteins and provide a defined environment to the cell, and also regulates transport of molecules in a selective manner. Eukaryotic cells possesses membrane bonded cell organelles. जो भी eukaryotic cells हैं उनमें कुछ membrane bonded cell organelles होती हैं जो कंट्रोल करती हैं exchange of essential component within the cell. And all the cell membranes are made up of glycerophospholipids. So जो भी cell membranes हैं उनकी जो basic composition है they are made up of glycerophospholipids. Basically जो glycerophospholipids हैं उनसे बना होता है. And plasma membrane is dynamic or fluid structure which forms exterior boundary of the cell and functions as a selectively permeable membrane. It means कि इसमें कुछ particular substances ही enter कर सकते हैं और exit कर सकते हैं. So that's why जो biological membranes होती हैं they all are selectively permeable membranes. If we talk about its composition, so in liver cells, plasma membrane consists of 54% lipids and 46% proteins, while in RBC membrane, it contains 43% lipids and 46% proteins by its weight. If we talk about its carbohydrate composition, so it is 5 to 10% of its total weight. So this is the basic information regarding plasma membrane. Next, plasma membrane helps in signal transduction. So it forms a very important barrier between the extracellular environment and the intracellular environment. जो भी signal transduction है, जो cell है वो sense करता है कोई भी environmental changes को. So the first receptor is cell membrane. So in cell membrane there are some particular receptors on the membrane जो external stimulus को receive करते हैं and they respond to that stimulus. So proteins found as receptors. There are some proteins which acts as receptors that receive extracellular stimulus and activates intracellular responses. तो ये कुछ एक ऐसे extracellular stimulus को respond करते हैं और cells को activate करते हैं to respond to those particular stimuli. Proteins make extracellular attachment sites for effectors. Effectors जो हैं they are stimulators basically, like we have hormones. ग्रोथ फैक्टर्स हैं जो इनिशिएट करते हैं वेरियस रिस्पॉन्सेज को सेल के अंदर. while we have some viruses like we have HIV, that is human immunovirus deficiency virus, that can change these receptors and gets entry into the cell. So HIV जो virus है ये receptors की conformation को change करता है and gets enter inside a cell. So this is the basic nature how it enters in the cell. Next comes models of cell membrane structure. So there are various models which were provided to explain the basic nature of cell membrane. Firstly, we have lipid and lipid bilayer model, then Danneli model, then we have Robertson urinary membrane model, and fluid mosaic model, which was given by Singer and Nicholson and is the most accepted model of plasma membrane. The first model of plasma membrane is the lipid bilayer model, which was proposed by Gorter and Grendel in 1925. For this, they used acetone to extract the lipids from the known quantity of RBCs. And after evaporating the solvent, जो solvent है उसको evaporate किया और उसके बाद उन्होंने measure किया area जो occupy किया है extracted lipids ने. the area that is occupied by the extracted lipids as a monomolecular film. As a monomolecular film at an air-water interface by using a method named as Langmuir-Truff. तो एक मेथड है जिसका नाम है Langmuir-Truff. इससे इन्होंने ये मेजर किया कि जो लिपिड्स हैं उनका जो एक्सट्रैक्टेड लिपिड्स है उनका एरिया कितना है as a monomolecular film. So from this study they observed that the area of the film of the extracted lipid and from the surface area of RBCs estimated by light microscopy, they made a conclusion that it is very clear that all our results fit in well with the supposition that the chromocytes are covered by a layer of fatty substance. जो RBCs हैं वो बाहर से covered हैं एक फैटी सब्सटेंस है जो एग्जिस्ट करता है 2 मॉलिक्यूल्स थिक और वो एग्जिस्ट करता है 2 मॉलिक्यूल थिक layers, means it is made up of lipid bilayer. So 2 layers हैं lipid की. So this was the first model proposed for explaining plasma membrane. Next comes Danneli and Davson model, which is also known as sandwich model. They have concluded that there are two protein layer जिनके बीच में जो lipid bilayer है that is sandwiched. So 2 protein layers के बीच में जो lipid bilayer that is sandwiched, and this model is known as sandwich model. So model explain biological and chemical properties of the membrane. And plasma membrane, according to their model, according to this model, that plasma membrane consists of 2 layers of phospholipid molecules in which phospholipid molecules are arranged with polar hydrophobic heads facing outside and hydrophobic non-polar lipid chains facing inside. So according to their model, जो लिपिड बाइलेयर्स हैं इनका जो हेड है वो बाहर की तरफ फेस करता है दोनों का, और जो टेल्स हैं दे फेसेस टुवर्ड्स इनसाइड, एंड सजेस्टेड दैट लिपिड मॉलिक्यूल्स आर अटैच्ड विद मोनोमॉलिक्यूलर लेयर ऑफ ग्लोबुलर प्रोटीन्स. सो ये अटैच होता है मोनोमॉलिक्यूलर लेयर ऑफ ग्लोबुलर प्रोटीन्स. And model was also called sandwich model as it displayed the lipid layer as sandwiched between the 2 protein layers. So ये lipid bilayer है ये sandwich है 2 protein layers के बीच में, जो 2 globular proteins हैं उनके बीच में sandwich. So that's why this model is known as sandwich model or Davson's Danneli model. It was given in 1935. Next comes Robertson's unit membrane model. According to this model, that the cell membrane is trilaminar structure and consists of 2 dark osmophilic layers. इसमें 2 dark हैं osmophilic layer separated by a light osmophilic layer. तो 2 dark osmophilic layers हैं separated by a light osmophilic layer. Thus, unit membrane was 75 Armstrong thick and 35 Armstrong thick phospholipid layer between the 2 20 Armstrong thick protein layers. So जो इसका size है that is 75 Armstrong और ये है sandwiched between 2 protein layers which are 20 Armstrong thick. Next comes the most accepted model of plasma membrane, that is fluid mosaic model, which was proposed by Singer and Nicholson in 1972. This is the photograph which shows Singer and Nicholson. It explains that molecular organization of lipids and protein in cell membrane. जो organization है molecular level पे lipids और proteins की cell membrane में, इन्होंने इसको explain किया. And the lipids and glycoproteins make roughly equal contribution. जो lipids हैं और glycoproteins हैं उनका जो ratio है that is almost equal in formation of membrane. Then lipid belongs to several classes. जो lipids हैं वो different classes को belong करती है, like we have phospholipids, glycerosylglycerides, galactosylglycerides, and sterols. These molecules share an important chemical property. इनकी कुछ common properties होती हैं. They are amphipathic, containing both hydrophilic, that is water-loving part, and a hydrophobic, that is water-fearing part. So जो lipids bilayer है इसका जो head है that is hydrophobic, और इसकी जो tails होती हैं they are long chain, they are made up of long chain of hydrocarbons जो कि हाइड्रोफोबिक होती है. Firstly, we'll discuss about the phospholipids. So these are the most common lipids which are present in the cell membrane and have a charged phosphate containing head and 2 hydrocarbon tails. So ये बना होता है एक phosphate head से जो इसको बनाता है हाइड्रोफिलिक, और 2 long chain fatty acids की tails जो होती हैं हाइड्रोफोबिक. So fatty acid tail contain between 14 to 24 carbon atoms. जो फैटी एसिड्स टेल हैं इसमें 14 से लेके 24 कार्बन एटम्स होते हैं एंड एटलीस्ट वन टेल हैज़ वन और मोर सिस डबल बॉन्ड। सो जो टेल है उसमें एक या एक से ज़्यादा डबल बॉन्ड हो सकते हैं। दैट ब्रिंग्स अनसैचुरेशन और आल्सो हेल्प्स इन मेंटेनिंग द फ्लूइड नेचर ऑफ मेम्ब्रेन। These double bond produces kinks and influence packing of the molecules, in turn affect the overall fluidity of the membrane. So जो ये fluidity है membrane की, that is affected by the presence of these double bonds. Phospholipids are of two different types. Firstly, we have glycerophospholipid. It is derived from glycerol, which is the most common type of lipids. It contains glycerol moiety, which means glycerol binding site, then fatty acids and alcohol containing substance, that is like we have choline and serine, and it also contains phosphate group. This is the basic structure of glycerophospholipid. It contains the glycerol group, in which one carbon contains this phosphate group, which forms this lipid hydrophilic, means this head portion hydrophilic in nature. Then with two oxygen or OH group, it is joined with the two fatty acids. जो fatty acids हैं ये बनाते हैं इसको hydrophobic, and these are known as hydrophobic tails. They are classified on the basis of alcohol moiety. जो glycerophospholipid है उनको classify करते हैं alcohol moiety के बेस पे, जो attach होती है phosphate group में, जो R है that represents the alcohol moiety. For example, we have choline. If attached with the phosphate group as an alcohol, the molecule is known as phosphatidylcholine. इसे कहा जाता है phosphatidylcholine और lecithin. So this is the chemical structure of lecithin. It consists of a choline head जो attach होता है phosphate group से. This phosphate is attached with this glycerol because glycerol contain a phosphate moiety. Then it contains two fatty acids जो join होते हैं glycerol group से. And next, what is lecithin? Lecithin is a protein which is used for treating memory disorders such as dementia, Alzheimer's disease. It is also used for treating gallbladder disease, liver disease, and certain types of depressions, high cholesterol, anxiety, and skin disease called eczema. So basically ये जो phosphatidylcholine है इसको artificially use किया जाता है to treat these diseases. If serine is attached to the phosphate group, अगर choline की जगह पे serine present है, which is an amino acid, then the molecule is called phosphatidylserine. तो इसे कहा जाता है phosphatidylserine. And if this next type is sphingophospholipids. It contains amino alcohol named sphingosine in place of ग्लिसरॉल. सो ग्लिसरॉल की जगह पे यहाँ पे प्रेजेंट होता है एक अमाइनो अल्कोहल जिसे बोलते हैं स्फिंगोसिन, एंड अ फैटी एसिड, फॉस्फेट ग्रुप एंड अल्कोहल लिंक्ड फॉस्फेट मॉइटी. और जो सारी चीज़ें हैं दैट इज ऑलमोस्ट सेम एज दैट ऑफ फॉस्फोलिपिड्स. सो स्फिंगोमाइलिन इज द मोस्ट कॉमन स्फिंगोफॉस्फोलिपिड विच इज़ प्रेजेंट इन नेचर. जो स्फिंगोमाइलिन है वो सबसे कॉमन जो Sphingophospholipids present है, that is sphingomyelin. And eukaryotic cell membrane is made up of 4 phospholipids. जो eukaryotic cell membrane है उसमें 4 phospholipids होते हैं. For example, we have phosphatidylcholine, phosphatidylserine, phosphatidylethanolamine and sphingomyelin. So ये 4 तरह के बेसिक जो phospholipids हैं ये present होते हैं eukaryotes के cell membrane में. So this is the structure which shows the phospholipids. So this is the glycerophospholipid in which it contains glycerol group which forms the backbone of this lipid, और जिसके साथ attached होता है phosphate group, and this phosphate is attached with the alcoholic moiety which contains either choline, ethanolamine, serine, inositol or glycerol, and contains 2 fatty acids. While sphingomyelin, which is sphingophospholipid, it contains sphingosine in place of ग्लिसरॉल. जो ग्लिसरॉल है उसकी जगह पे स्फिंगोसिन प्रेजेंट होता है विच फॉर्म्स द बैकबोन ऑफ दिस लिपिड, और इसमें अटैच होता है फॉस्फेट ग्रुप विच इज अटैच्ड टू द alcohol moiety, for example choline, and it also contains 2 fatty acids. So there are basically 2 types of phospholipids. One is glycerophospholipid. Next comes glycolipids. As the name indicates, glycose means sugar, lipids means fats. So it contains sugars which are attached with the lipids with the help of covalent bonds. So this is the lipid bilayer, और इसके साथ attached हैं ये sugars. दे मे बी सिंगल शुगर ग्रुप और मे बी फ्यू शुगर ग्रुप्स. Oligosaccharide भी हो सकते हैं, monosaccharide भी होते हैं, which are joined with lipids with the help of covalent bonds. So found on the exterior side of the membrane, जो बाहर की साइड है मेम्ब्रेन के उसमें प्रेजेंट होते हैं. जो कार्बोहाइड्रेट्स प्रेजेंट है दे में मोनो और ओलिगोसैकेराइड्स which are covalently bonded to the lipid moiety. And most common glycolipid is cerebroside. सबसे कॉमन ग्लाइकोलिपिड प्रेजेंट होता है प्लाज्मा मेम्ब्रेन में, दैट इज सेरेब्रोसाइड. इट कंटेन्स सिंगल शुगर, आइदर इट इज ग्लूकोज और गैलेक्टोज. सो इसमें जो शुगर प्रेजेंट होता है या तो ये ग्लूकोज प्रेजेंट हो सकता है या फिर गैलेक्टोज. सो दिस इज द बेसिक केमिकल स्ट्रक्चर ऑफ दिस cerebroside. It contains sphingosine, then it contains long chain fatty acids and contain one sugar that may be galactose or glucose, and this is the most commonly occurring glycolipid present in the cell membrane. Then it contains second class, that is gangliosides. As the name indicate, they are the more composite glycolipids comprising of branched chain of carbohydrates. So इसमें एक से ज़्यादा जो sugar groups हैं वो present होते हैं. These are known as gangliosides. So this photograph shows cerebroside, which contains single sugar group that may be either glucose or galactose, while ganglioside contains branched chain of carbohydrates. Next comes sterols. Sterols का अगर chemical composition की बात करते हैं, it consists of its steroid nucleus. इसमें एक present होता है steroid nucleus containing 4 fused rings, और इसमें होती हैं 4 fused rings, 3 with 6 carbons. जो 3 है first 3 they are with 6 carbons and the 1 with 5 carbon, और जो last वाली है उसमें होते हैं 5 carbon. So this is the basic structure of sterols, which consists of its steroid nucleus containing 4 fused rings, जो उसमें से 3 है with 6 carbons and 1 is with 5 carbons. And the most common sterol is cholesterol and is most commonly present in the animal cells. जो ये स्टेरॉल है ये एनिमल सेल्स में प्रेजेंट होता है, प्लांट सेल में इट इज नॉट प्रेजेंट. देयर आर अदर टाइप्स ऑफ स्टेरॉल्स. प्लांट कंटेन्स अदर स्टेरॉल्स लाइक स्टिग्मा स्टेरॉल और साइटोस्टेरॉल्स. देन बैक्टीरियल सेल आल्सो लैक्स कोलेस्ट्रॉल. बैक्टीरिया के सेल में भी कोलेस्ट्रॉल एब्सेंट होता है एक्सेप्ट माइकोप्लाज्मा. जो माइकोप्लाज्मा है या पीपीएलओ जिनको बोलते हैं, प्लूरोनिमोनिया लाइक ऑर्गेनिजम्स, उनमें प्रेजेंट होता है कोलेस्ट्रॉल. Lipids like cholesterol or sphingolipids are aggregated to form lipid rafts. जो एक और स्ट्रक्चर है जिसे कहा जाता है लिपिड राफ्ट्स. सो ये बनता है कोलेस्ट्रॉल या स्फिंगोलिपिड्स जो एग्रीगेट होते हैं, दे विल गिव राइज टू दिस स्ट्रक्चर दैट इज लिपिड राफ्ट. Basically, the plasma membrane of cell contain combinations of glycosphingolipids, cholesterol and protein receptors. So ये जो combinations है which contain glycosphingolipids, cholesterol और protein receptor, they organized in glycolipoprotein lipid microdomains, जो कि organize करके बनाते हैं glycolipoprotein lipid microdomains, जिन्हें कहा जाता है lipid rafts. So ये जो सारे के सारे structures हैं they combine or aggregate to form this structure which is known as lipid rafts. Then heterogeneous in nature, contains both protein as well as lipids. और जो lipid rafts होते हैं they are heterogeneous in nature, means they are made up of 2 different types of substances, like it contains both proteins as well as lipids. And main function like cholesterol trafficking, और जो lipid rafts का function है it is mainly involved in cholesterol trafficking, then signal transduction में help करता है, and endocytosis. That is, next comes fluidity of membrane. So this dynamic nature of a membrane is due to the movement of these lipids molecules either along the axis of lipid bilayer or on lateral position. So lipid bilayer can either move along their axis, या तो ये अपनी axis पे move कर सकते हैं like from this axis to this one, and this is called rotational movement, or we can say this is flip-flop movement. Second, they can laterally diffuse, वो laterally diffuse कर सकते हैं from this position to the other position on the lateral side, and this is called lateral motion. So lipid to diffuse along the side in lipid bilayer signifies the fluid-like properties of lipid in the cell membrane. So ये जो lateral movement है lipids की, ये indicate करता है fluid nature of this membrane. The lateral movement can be visualized through fluorescence microscope. अगर हमने ये जो movement है ये देखनी है, so we can use a device known as fluorescence microscope by a technique named as FRAP. and it stands for fluorescence recovery after photo bleaching. So इस टेक्निक से हम इनकी मोशन को चेक कर सकते हैं या ऑब्जर्व कर सकते हैं. This movement from one side of the leaflet to the another is known as flip-flop motion. जो ये lipids की movement है इस one leaflet से दूसरे leaflet में, this movement is known as flip-flop motion. This fluidity of bilayer depends on the composition of lipid molecules and external temperature. So दो factors हैं जो fluidity को affect करते हैं. One is lipid molecule, second is external temperature. So decrease in outer temperature results in reduction in the membrane fluidity. अगर temperature कम हो जाए तो जो fats हैं वो solidify हो जाते हैं. So it results in membrane fluidity reduction and form rigid gel-like structure, जिससे कि rigid gel-like structure बनता है. A response of lipid bilayer towards the temperature change is defined as phase transition. So जो ये temperature की fluctuation के साथ lipid की जो composition है वो change होती है उसे कहते हैं phase transition, and temperature at which transition takes place, और ऐसा temperature जिसके ऊपर this transition takes place, this is known as transition temperature. इसे कहते हैं transition temperature. And fluidity also depends on the length of the carbon chain and degree of unsaturation. So जो और factor है responsible fluidity के लिए, एक तो length of carbon chain, जितनी ज़्यादा लंबी chain होगी, और दूसरा degree of unsaturation. So it also affects the fluidity. So lipids with short carbon chains and less unsaturated fatty acid residue experiences the transition at lower temperature as compared to the lipids with long or saturated fatty acyl chains. It means जो lipids हैं which are shorter, which containing shorter carbon chains और unsaturated fatty acids, तो ये ऐसे जो lipids हैं वो कम temperature में भी changes को show करते हैं. Means they respond to the external temperature change, while those lipids which contain longer carbon chains और जिसमें saturation ज़्यादा है, means they contain single bonds, उनमें जो temperature fluctuations है वो less होती है with the change in temperature. This was all about for our today's discussion about cell membrane. In next part of this topic, cell membrane, I will cover the membrane proteins and translocation through the membrane. If you have any questions, queries, and any suggestions, you can give it in comment section. Thanks for watching. Have a great day.